In this video, we will discuss about the different generation of chimeric antigen receptors and some of the logics that they used while designing those chimeric antigen receptors. So, we discussed there are several generations of the chimeric antigen receptors previously. So we will start with the first generation CAR, which only has this CD3 intercellular domain. Apart from that, they has this transmembrane domain, extracellular domains, and also this hinge that connects the extracellular domains to the transmembrane domains. And once these extracellular domains interact with the TA, that is tumor associated antigens, it will trigger the activations of the CD3 or the CD3. After the CD3 intercellular CD3 signalings will cause the activations of the T cells, and here there is no co-stimulatory domains. So this is just a first generation CAR, and here the effect of this CAR would be suboptimal. And in the second generation CAR, they have used co-stimulatory domains, either CD28 or 41BB. In that case, the T cells not only get activated, but it will also cause the activations of the co-stimulatory pathways that will cause the release of the cytokines. At the same time, it will trigger apoptosis into the tumors or into the tumors that are detected by this CAR. And in the second generations, the only one of the co-stimulatory domains used at a time, either CD28 or 41BB. But in the third generation CAR, both the co-stimulatory domains can be used all in the same plasmid or in the same cells. That is, CD28 and 41BB are present in the same plasmid and along with the CD3. This is the third generation CAR. And in the fourth generation CAR, they have introduced another this stat binding motif that can helps in the cytokine release or immune checkpoint inhibitors, or prevent these immune checkpoint inhibitors that are present on the tumor cells to deactivate the T cells. This is the fourth generation CAR. And in the fifth generation CAR, we are using one of this interleukin 2R beta also, and this along with the other co-stimulatory domains will enhance the activity of the T cells. So superactivities or superactivated T cells are also not desirable, as it might cause undesirable reactions or undesirable results or non-specific killing of the cells. Now let us discuss what are the conditions or some of the gates that are being used while designing this CAR. One of the gates is that if the two different cancer antigens are found out, then the bispecific antibodies can be used. In that case, when both the antigens are present on the cancer cells, then only it will trigger the immune cell activations. Here the AND gate is used, that is both the antigen needs to be present. In the other case, if the normal antigen is present and the cancer antigens are also present, then in that case it will use AND NOT gate. That is, when both of them are present, then there is a possibility that it could be healthy cells, then it will not trigger immune cell activations. If only, and in the third case is that when both the antigens are detected, then it can trigger immune cell activations in either of the cases. That is, when either antigen 1 is detected or antigen 2 is detected. When any one of them is detected, then it will trigger immune cell activations. So these are some of the, and this is used when the antigens are more specific to the cancer cells. So these are some of the brief overview about different generations of the CAR and what are the different kind of gates that can be used, or logic can be used while generating or while developing these cars. If you need to understand more about this uh, car in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.